Good day, everyone. Today, we are going to present a research study entitled Assessing the Effects of Probiotics to Growth and Yield of Rice, Oryza sativa. We are the presenters of this research study. I am RGC Imag. We are from the Bachelor of Secondary Education major in Science of the College of Education at Jose Rizal Memorial State University, Dipolog Campus, Dipolog City. The Philippines agricultural sector has declined in previous years. In fact, according to Briones 2021, there are several reasons as to why there is a decline of the Philippines agricultural sector, and these are reduced farm sizes, low salaries, restrict access to funding, inadequate infrastructure, harsh weather, population increase on rural areas have contributed to low agricultural productivity and have forced people out of the industry. In addition, the Department of Agriculture 2022 reported that COVID-19 pandemic, the African swine fever, and the conflicts between Russia and Ukraine have severely damaged the agricultural industry. However, it began to recover in the year 2022 according to Statista Research Department 2023, although a bit slower in comparison to the pre-pandemic periods. It contributed 9.1% of the nation's GDP, a rise of 2.2% in the previous year. In fact, the recommended budget for the Department of Agriculture in the year 2024 is approximately 197.84 billion pesos. According to Mahmoud et al. 2023, most issues caused by overusage of synthetic fertilizers can be resolved by using greener fertilizers such as biofertilizers, which are also based on plant probiotic bacteria. Microorganisms known as plant probiotic can colonize plant roots and encourage plant growth via a direct and indirect method, enhancing the plant nutritional content and crop quality, according to Gomez et al. 2017. According to Ramananzang 2018, crop yields are lower as a result of the substantial environmental damage caused by overusage of synthetic fertilizer. In addition, Kumar and Kishar 2017 stated that excessive utilization of synthetic fertilizer leads to pollution, particularly water pollution, as the main contributors to it are the phosphates and nitrates present in synthetic fertilizer. Additional issues with synthetic fertilizer include decreased farmland diversity, low soil nutrient level, genetic diversity loss, and increased greenhouse gas emission, according to Fraile and Mendez 2017. In fact, according to Statista Research Department, approximately 15.4 million metric tons of rice were consumed in the Philippines in the year 2022 to 2023, a rise from previous years. According to Don et al. 2022, one of the world's top rice-consuming nations is the Philippines. To address such major demand, enhancing the relationship between rice plant and the microbiome that lives on, inside, and around them is one method to further increase rice output. Hence, this study aims to assess the significant effects of the application of probiotics to growth and yield of rice Oryza sativa variant 222 in three major groups. One group treated with probiotic lactoserum only, one group treated with synthetic fertilizer NPK only, and one control group that is untreated with both probiotics and synthetic fertilizer in a well-drained and undrained soil.
And I am Josemay Alaga Sulamilio. Now, let's proceed to the research objectives. At the end of this research study, the researchers will be able to assess the following. First, the effects of probiotics lactoserum to the growth of rice Oryza sativa variant 222 in terms of plant height in centimeter, number of leaves, length of leaves in centimeter, number of tillers, and number of effective tillers. Second, the effects of probiotics lactoserum to the yield of rice Oryza sativa variant 222 in terms of filled and unfilled grains, number of rice stalks, length of rice stalks, and weight of the harvest. Third, the effects of the application of synthetic fertilizer NPK to the growth of rice Oryza sativa variant 222 in terms of plant height, number of leaves, length of leaves, number of tillers, and number of effective tillers. Fourth, the effect of the application of synthetic fertilizer NPK to the yield of rice Oryza sativa variant 222 in terms of filled and unfilled grains, number of rice stalks, length of rice stalk, and weight of the harvest. Fifth, If there is a significant difference in the growth and yield of rice or Rhiza sativa variant 222 between the well-drained and undrained rice plants. And the last one is if there is a significant difference in the growth and yield of rice or Rhiza sativa variant 222 between three groups. One group treated with probiotics lactoserum only, one group treated with synthetic fertilizer NPK only, and one control group that is untreated with both probiotics and synthetic fertilizer respectively.
Good day, dear panelists. I am Mr. Philip J. Zhao, and now let's proceed to the theoretical or conceptual framework of the study. This study is anchored to Mechnikov's theory that was introduced by Elie Mechnikov in 1907. So this theory covers different scientific investigations such as phagocytosis, cellular innate immunity, gerontology, and most especially probiotics. So Mechnikov found out that the people who live in Kusakos Mountains, part of Bulgaria, were regularly consuming a fermented yogurt beverage that has a Lactobacillus bulgaricus in it. So it is a type of probiotics that appears to enhance their lifespan and general health. This current research is similar to the mentioned theory since both are focusing and investigating about the significant effect of using a probiotic to a specific subject. Moreover, this study is also anchored to research done by Islam T et al. 2023 entitled Improvement of Growth, Yield and Associated Bacterium of Rice by the Application of Probiotic Parabucalderia and Delphtia. This research focused on evaluating about the impact of applying these probiotics to rice roots, rhizosphere soil bacterium, the growth, the yield, and to the different NPK fertilizer dosages. So the probiotics that they used in this study were the Parabucalderia fungorum strain or the BRRH4, the Delphtia sp strain, the BTLM2, and also they added some chemical fertilizers on it. Now, moving on to the research method. So method is very important because this will serve as the guide and the basis for the researchers in conducting the experiment. First method is the preparation of rice seeds, probiotics, and materials. It is very easy and convenient for the researchers to prepare these materials since the variable of the study are abundant in the locale. And there's only one variety of rice that will be utilized in this study, and this is the variant triple 2, which will be collected in the City Agriculturalist Office located at Biaso Dipolog City. Meanwhile, the other materials that will be utilized in this study are the following. We have the petri dishes, the filtered paper, the gloves, the graduated cylinder, the weighing scale, the pH meter, the meter stick, the hose, the rake, the grape hoe, the grass shear, the wood or bamboo stick, nails, recyclable tarpaulin, rope, styrofoam boxes, socks, shovel, and synthetic fertilizer or the NPK, and some of the materials will be purchased, some of the materials will be collected in the researchers' houses, and some of the materials will be borrowed into the science laboratory of Ocerizal Memorial State University, Dipolog campus. The probiotic lactoserum that will be utilized in this study will purchase in the probiotic company located at the Pitan City. Now, the researchers will gonna identify of which type of bacteria is the best to use in order to see the effectiveness and survival during the inoculation process. The second method is measuring probiotics and weighing of synthetic fertilizer. In this study, researchers will no longer culture the bacteria that will be used as growth and yield agents for the rice or Rhizosativa variant triple 2, as they will be directly purchased from the probiotics company in the Pitan City. Likewise, synthetic fertilizer which is 14-14-14 NPK will be bought online. Meanwhile, researchers will measure the amount of probiotics in milliliters, which will be converted to percentage to be applied to the rice seeds during inoculation. Specifically, the desired measurement for the probiotics per petri dish is 25 ml, and this measurement is equivalent to 25% of 1000 ml probiotics. It is enough to cater 25 surface sterilized rice seeds. On the other hand, the ideal weight of the synthetic fertilizer that will be applied to the rice seedlings per application is 187.5 grams, which is equivalent to 37.5% of 1,500 grams NPK in total. The measurement of probiotics and the weight of the synthetic fertilizer will be obtained by using a graduated cylinder, and a weighing scale, respectively.
So now let's proceed to the next method, which is the inoculation and germination of rice seeds. After preparing the synthetic fertilizer, probiotics along with the rice seeds, inoculation will then follow. For the controlled group treated with probiotics only, 25 surface sterilized rice seeds will be soaked in 25 ml or 2.5% of probiotics. Then, the rice seeds treated with probiotics will be placed on a petri dish containing a filter paper that will serve as a medium for the seeds during germination. To obtain a better bacterial coating, the rice seeds treated with probiotics will be then let to dry overnight at room temperature. Watering will occur every other day according to Mahanta et al. 2017. Furthermore, the rice seeds will be given 5 days for the inoculation and germination on a petri dish. Then, it will be examined. On the percentage or in the germination assay test, the germination percentage will be examined or determined once at 3 days after inoculation. Then, other parameters will be measured such as the height of the seedlings, the length of the leaves, and the number of leaves. This will be conducted in the Science Laboratory at the Sarazal Memorial State University, Dipolog Campus, Dipolog City. On the other hand, the remaining 2 groups of rice seeds, untreated with probiotics, of course, will be directly grown on a styrofoam box that will serve as the seedbed within 25 days prior to transplanting on the rice plots. So now let's proceed to the next method, which is the seedbed preparation and transplanting. The researchers will prepare 6 seed beds, 2 for the group treated with synthetic fertilizer only, 2 for the group treated with probiotics only, and 2 for the control group that is untreated with both probiotics and synthetic fertilizers. Researchers will employ recycling process as the main materials to be used in this study for the seedbed are the styrofoam boxes that will be collected from the community. 3 styrofoam boxes for the well-drained soil and 3 styrofoam boxes for the undrained soil, both having an area of 10 by 10 square centimeters. Researchers will prepare a dark gray loam type of soil with a pH level of 5.5 and a height of 100 centimeters. The soil will be flattened and a drainage canal will be created to remove some of the waters in the seedbed for better growth of the rice seedlings. The seeds treated with synthetic fertilizers and for the control group that is untreated with both probiotics and synthetic fertilizers will be directly grown on the seedbed. Meanwhile, the rice seeds treated with probiotics during inoculation will be transplanted on the seedbed after inoculation. Then all of the rice seedlings will be grown in the seedbed for 25 days prior to transplanting on the rice plots.
The next method is preparing for the rice plots. The researcher will prepare 6 rice plots with an area of 5 by 5 square meter. Spacing per seedling is 20 centimeter. 3 plots will have well-drained soil, while the other 3 plots will have undrained soil, both having a height of 100 centimeter. For the undrained rice plots, the appropriate surface level of water from the soil is 2 to 3 centimeter, and 2 rice plots will be assigned per group. 1 group treated with probiotics lactoserum only, and 1 group treated with synthetic fertilizer or NPK only, and 1 control group that is untreated with both probiotics and synthetic fertilizer. And each group will have a multiple replications. And this will be prepared in the rice fields located at Tubod Galas, Dipolog City. And in order to successfully initiate the desired rice plot, materials such as grass shears, rake, grape hoe, sacks, shovel, and hose will be used. And also appropriate type and pH level in soil will take into considerations, in which for the cultivation of rice Oryza sativa variant 222, a dark gray loam soil type with a pH of 5.5 is preferred, according to Kumar et al. 2019. And successfully preparing the rice plots will ensure the survival of the rice seedlings. The next method is transplanting rice seedlings. The seedlings that will be grown on the seedbed for 25 days will then be transplanted to the rice plots prepared by the researchers. Meanwhile, the researchers will again prepare 1000 ml or 100% of probiotics mixed with sterile distilled water of 500 ml. And in order to improve the bacterial root colonization for those seedlings treated with probiotics during inoculation, researchers will wash them, and their roots will be immersed in the probiotic sterile distilled water solution for an entire night prior to transplanting on the rice plots. In carefully prepared rice plots, 25-day-old seedlings will be transplanted. And the 3 rice plots will, for the both well-drained and undrained soil, will have 564 rice seedlings each, a total of 1,128 rice seedlings. And for the both well-drained and undrained soil, 188 seedlings will be for the group treated with probiotics lactoserum only, 188 rice seedlings for those group treated with synthetic fertilizer or NPK only, and lastly 188 seedlings for the control group that is untreated with both probiotics and synthetic fertilizer. And there will be multiple replications per group. After transplanting, the seedlings' growth will be monitored for up to 3 months prior to harvesting.
Now, let's proceed to the next method, which is the application of synthetic fertilizer on the rice seedlings. The researchers will apply synthetic fertilizer NPK to the second controlled group, treated with synthetic fertilizer only. Each plot in this group will receive 187.5 or 12.5% of synthetic fertilizer per application period. The prepared 187.5 or 12.5% of synthetic fertilizer will be applied 3 times on the rice seedlings. Once 12 days after transplanting, once during the tillering stage 25 to 30 days after transplanting, and once at wooding stage 40 to 45 days after transplantation. The rice seedlings will then be monitored by the researchers to ensure that there are no interruptions in their growth. The last method is the harvesting and collection of data. The rice plant among the 3 groups, 1 group treated with probiotics lactoserum only, 1 group treated with synthetic fertilizer NPK only, and 1 control group that is untreated with both probiotics and synthetic fertilizer will be harvested after 3 months of its growth phase. Researchers will record the data every after 15 days while the rice seedlings are still growing. Meanwhile, filled and unfilled grains, number of rice stalk, length of rice stalk, and weight of the harvest are for the yield of rice. The data to be collected will be analyzed using statistical tools.
So this is the flowchart of the research methods.
The proposed budget for this study, 10 bottles of probiotics will be cost 2,000 pesos, 25 kg of synthetic fertilizer NPK it will be cost 1,625 pesos, and for the rice plot it will be cost 9,000 pesos, and for the hose 2,000 pesos, and for the fair 7,200 pesos, and for the others 8,000 pesos, a total of 29,825 pesos.